All right. Hi, everyone. I'm Samara Zinman. I'm a current master's student at Nova Southeastern. As he said, I'm going to be looking at talking about the geographically driven differences in the microbiomes of staghorn corals originating from across Florida's coral reef. So in order to have successful restoration, we want to consider the whole coral holobiont. So not just the coral host, but the symbiotic algae and the microbiome as well. And this plot's a great representation of the relationship between the different aspects of the holobiont. And as you can see, there's a lot going on the microbiome side of the plot because the microbiome provides a lot of different services to its coral host, such as it can affect disease resistance. So I know Courtney already talked about this study a little bit, and you can go see Grace talk about it in the disease session after this, but a study done in 2020 looked at the microbiomes associated with different genotypes of staghorn corals that were identified as susceptible or resistant to disease. And as you can see in the susceptible genotypes, they found pretty much across the board, they were all dominated by one bacteria, the bacteria in green, Aquarickettsia, whereas the disease-resistant genotypes had little to no Aquarickettsia at all, and these disease-resistant genotypes are really rare, so we want to know as much about them as possible. And given this link between Aquarickettsia and disease-susceptible staghorn corals, investigating staghorn coral microbiomes across the Keys could give us insight into potential geographic associations of Aquarickettsia and disease-resistant staghorn corals, which could potentially be harnessed for restoration purposes. So microbiome composition is affected by many sources, things like the host species, the region, and the environmental conditions. So it's important to consider the environmental conditions of Florida's coral reef before proceeding. So due to variation in the ocean circulation patterns, Florida's coral reef has regional differences driven by the passes and tidal channels flowing from the Gulf of Mexico and Florida Bay, which provides variable environmental conditions for the middle and lower Keys that the upper Keys are protected against. And in terms of what this means for restoration corals could vary in their microbiomes based on their original source reef or their outplanting location or nursery location. Leads to our research goals. So we want to examine the longevity of a regional signal in staghorn coral microbiomes and assess the relationship between Aquarickettsia and region, and compare composition and diversity across restoration staghorn coral genotypes. And this project actually came about because we had a bunch of corals that originated in the source reefs at the various points there across Florida's coral reef before they were all collected and integrated into Mote Marine Laboratories in situ nursery at the Gold Star in the lower Keys there. They lived together in that nursery for several years before we did any microbiome sampling. And because of this, it gives us a really unique opportunity to look at potential regional signals in restoration staghorn coral microbiomes. So between November and December 2019, we collected 74 staghorn coral genotypes from Mote's in situ nursery. We had 40 genotypes that originated from the lower Keys and resided in Mote's nursery for about 8 years. We had 15 that originated from the middle Keys and resided in Mote's nursery for about 3 years, and 19 from the upper Keys that resided in Mote's nursery for, again, about 3 years. In terms of our sample and data processing, we took tissue scrapes of our genotypes, which we then performed DNA extraction on and sent off to Mr. DNA for 16S rRNA sequencing, which provided us with sequence data that we could then feed into the Data2 pipeline and Silva database to get our amplicon sequence variant and taxonomy information, which we could then filter by prevalence to remove the taxa that were basically not present. So this leads to our results. We characterized the microbiomes of our staghorn corals in a relative abundance plot, and as you can see across all of the initial collection regions, the majority of the genotypes were dominated by one bacteria, just like what we talked about earlier, Aquarickettsia, the green color. However, there are a select few odd outlier genotypes that had little to no Aquarickettsia at all, and this lack of Aquarickettsia appears to result in visibly higher diversity and the dominance of Spirochaeta, which is the pink color, and an unclassified ASV that's in light blue. Now, for clarity purposes, for the rest of the presentation, I'm going to be calling the non-outlier genotypes high Aquarickettsia genotypes, and the outlier genotypes, the ones in the boxes, I'll call low Aquarickettsia genotypes. Now, given the role that Acropora and Spirochaeta, given the dominance of them in our corals, I just want to briefly go over the roles they play. So Acropora is a presumed coral parasite that leads to reduced growth rates. It steals ATP and amino acids from its coral host. Whereas Spirochaeta is found actually in healthy elkhorn corals and reduces with disease, and we think it's involved in coral nutrient cycling. So we then wanted to compare bacterial diversity among the 3 regions of the Keys, and we started with beta diversity, which remember is the diversity between groups. We used non-metric multidimensional scaling analyses with Bray-Curtis distances to look at the similarities among our genotypes based on by their initial collection region to produce this plot here. And alongside this NMDS plot, we also ran a permutational multivariate analysis of variance, which is a really unnecessarily long way of saying permanova. And so our permanova on our data, on the full data, it found significant differences by initial collection region. However, that's really hard to tell on our NMDS plot here because we had those low Acropora genotypes clouding things a bit. So we decided to get a clearer look by looking at our high Acropora genotypes and low Acropora genotypes separately. So when we looked at our high Acropora genotypes, our permanova still found a statistically significant difference by initial collection region, which is backed up by our NMDS plot that shows each region individual from each other. And you can see in our NMDS plot, we've got this transition from the lower Keys to the middle Keys to the upper Keys. So when we looked at our low Acropora genotypes, we found a significant difference by initial collection region, which is backed up by again by our NMDS plot. So we decided to further back up our beta diversity findings with the beta disper function in R, which calculates the multivariate homogeneity of dispersion of our Bray-Curtis distances. So basically, it looks at dispersion or the variation in microbiomes. So the more dispersed, the higher the dispersion, the bigger the diversity of microbiomes for that group, which is important because higher diversity is often linked with higher resilience. So when we looked at our high Acropora genotypes, we found significant differences between the lower and upper Keys, but we did not find significance between the middle Keys and either of the other regions. So this is really supporting what I had mentioned with our NMDS plot, where we had that transition from the lower Keys to the middle Keys to the upper Keys, and the upper Keys have the most dispersion, the most beta diversity. So we then looked at our low Acropora genotypes, and despite the fact that our permanova and NMDS both suggested significant differences by initial collection region, our beta disper analysis did not find that, but that's likely due to a low sample size as we didn't have a lot of samples here, and you can see there's a difference between the dispersion for the different groups in our plot here. I think it's also worth noting that the low Acropora genotypes are also much higher in beta diversity in general than our high Acropora genotypes. So you could just tell by the y-axis, which is our dispersion, it's just so much bigger for our low Acropora. So we then looked at our alpha diversity, the diversity within a group, and this is important to look at because we want more diverse bacterial communities in our corals. We looked at 2 metrics of alpha diversity. We looked at species richness, which is the number of taxa, and we looked at Shannon diversity, which is the number of taxa weighted by evenness. So for our high Acropora genotypes, our results were very similar to beta diversity. For both metrics, we found significant differences by initial collection region specifically between the lower Keys and the upper Keys. And for Shannon diversity, we found that the middle Keys had no significance between either region. So we're seeing the same kind of thing that we saw with beta diversity, where we have that clear transition there from the lower Keys to the middle Keys to the upper Keys. So when we looked at our low Acropora genotypes, we actually found no significance by initial collection region. We did, however, find that our low Acropora genotypes are significantly higher in alpha diversity than our high Acropora genotypes. So basically what this comes down to is you remove Acropora and alpha and beta diversity appear to increase. So just to wrap things up a bit, we found significant differences in the bacterial diversity of our genotypes driven by initial collection region, even after they are housed together in Mote's nursery for several years. And this supports the existence of geographic influences on host microbe interactions, which could be retained for years after relocation. But I don't really want to separate out what specifically is the environmental differences versus genotypic, because staghorn corals also vary by genotype based on their region. So we also found two key bacterial genuses, Acropora and Spirocheta, that may play an important role in distinguishing staghorn coral bacterial communities. And we found those genotypes, a few genotypes in the lower and upper keys that had low Acropora, which appeared to result in higher diversity. And this higher diversity has been proposed to be indicative of disease resistance, because the higher diversity takes up niche space that could otherwise be used by opportunistic species. So this is great, but what does it have to do with restoration? So given the important role some bacteria play for their coral hosts, which can result in varying resiliencies, understanding how microbiome composition and diversity varies by region can allow us to make informed decisions on which corals to plant based on their microbiome resiliencies. Additionally, genotype, environmental factors, and microbes have considerable influence on coral health and survival, so geographic differences in their initial collection region should be considered when determining outplanting strategies. And those low Acropora genotypes are more likely to succeed and survive in stressful environments and should therefore be thoughtfully targeted for restoration efforts. So I want to thank everyone who helped out with this project and take questions if we have time.
Thank you, Samara. Yeah, we have time for a couple of questions.
Yeah.
I was wondering, I might have missed it, but when you were talking about your collection of your samples, did you pull these samples originally from these different areas, brought them to a nursery for an amount of time, and then sampled from that nursery? Is that?
Yeah, so our corals were originally collected from those, you saw the map earlier,
Yeah.
but from the different source reefs, and then they were brought to the local nurseries there, and then throughout the various gene swaps across the years, they found their way to Mote. And then eventually we took tissue scrapes for microbiome analysis and went, that's weird.
And if I can ask one more follow-up, do you think maybe their time spent all collaboratively in this one nursery might have affected that microbiome?
So that's a great question and something we've extensively discussed, and that's hard to tell because we didn't get a chance to sample them right as they were taken off of their source reef. It's something we hope to study in the future. It's the next step we'd like to take, funding dependent.
Yeah.
Yeah.
I probably missed this, but when you were talking about how you processed your data, did you mention that you took out bacteria that were less than a certain percentage? Did I get that right?
Yeah, we had some bacteria that had like 0.0001% pop in, and our data just wasn't running that way. It kept crashing our computers. And
So what was your cutoff for that?
I have it in here because it's 10%, I think. I have it written in my thing somewhere. It's also in our paper, which I'm happy to send to you if you prefer.
Thank you.
Yeah.
Great, thanks, Samara. Our final presentation